Hi, everyone. This is the mid-semester 1 revision video, and I'll start by the first question, which is justify skeletal muscles are considered organs. A skeletal muscle like the biceps or the triceps muscle, for example, is considered an organ because it consists of a group of muscle tissues or a group of tissues, that carry out one or more related functions. So in case of the biceps muscle, for example, we have the muscular tissue that contracts and relaxes. We have the vascular tissue that consists of the arteries and veins and blood capillaries related to the biceps muscle, the arteries that carry the oxygen and nutrients to the muscle, the veins that carry the waste substances away from the muscle, as well as the blood capillaries inside the muscular tissue where exchange of material occurs. In addition to the skeletal muscular tissue, to the vascular tissue, we have the nervous tissue that consists of the motor nerve supply to the biceps muscle, the sensory nerve supply to the biceps muscle, as well as the myoneural junction, which is the synapse between the individual nerve fibers and the corresponding muscle fiber. And we also have the connective tissue that represents the fourth type of tissues. For example, in case of the biceps muscle, we have the epimysium that covers the whole muscle. And then we have the perimysium that surrounds the bundles. We have the endomysium that is present in between the individual myofibers. The second question that you might see on the test, or the second type of question, is to enumerate the different types of muscular tissue. So we have the skeletal muscular tissue that is by definition attached to the skeleton or to the bones. We have the smooth muscular tissue that lies mainly in the internal organs. And we have, of course, the cardiac muscle tissue that lies, or the myocardium that lies in the substance of the heart. You might also be asked about the definition of a muscle fiber. Actually, the muscle fiber is an elongated cell. So it is an elongated muscle cell. Muscle cells are called muscle fibers or myofibers because they are elongated. The skeletal muscles are elongated and cylindrical. The smooth muscles are a bit elongated, not to the extent of the skeletal muscles, but they are characterized by being tapered from both sides. The predominant characteristic, what is characteristic of all types of muscles, whether they are skeletal or smooth or cardiac, is their contractility or their ability to contract. And if you are ever asked to justify the ability of the muscular tissue to contract, you will definitely talk about the two important myofilaments in the sarcoplasm of any type of muscle, which are the thick myosin filaments and the thin actin filaments. In case of what's the difference between actin and myosin in smooth muscles and actin and myosin in skeletal or cardiac muscles? The main difference is in the arrangement. Is it a regular arrangement or an irregular arrangement? In case of the skeletal muscles and the cardiac muscles, the thick myosin and thin actin filaments are regularly arranged in an alternating pattern of dark bands where myosin and actin are present, and light bands where only actin is present. That's why the skeletal muscles and the cardiac muscles appear banded or striated under the light microscope and of course under the electron microscope. So if I ever ask you about the cause of banding or striation, you would talk about the regular alternating arrangement of myosin and actin. But in case of the smooth muscles, it's true that they can contract because of actin and myosin, but they are unstriated, they are unbanded. This is because there is no regular arrangement of actin and myosin. And now we're going to talk about the muscle functions. First of all, before talking about the muscle functions, we have to identify their location. Where is each of the following type of muscles located? As we said before, we have three types of muscles. The skeletal muscles, their location is attached to bones. The smooth muscles, they are mainly in the gut, in the internal viscera of the body, like for example, in the alimentary tract or in the urinary system, in the ureter, in the urinary bladder, and in the urethra, as well as in the eye, because we have seen that they are present in the iris of the eye. Smooth muscles are also present in the walls of the blood vessels. And therefore, if you want to talk about the location of the smooth muscles, it's not only one location, but different locations in the body. But in order to remember them, you have to remind yourself that all the locations where the smooth muscles are present are sites where you cannot control the muscle. For example, in the alimentary tract, in the urinary system, in the blood vessel wall, in the iris of the eye. And therefore, when you are to describe the functions of the smooth muscles, you have to differentiate between the different locations, meaning that the muscles in the smooth muscles in the gut have a main function, which is peristalsis. And peristalsis is important to sweep the food downwards. So every organ carries out peristalsis in order to sweep the food downward towards the organ that lies below it. For example, the esophagus carries out peristalsis to push the food to the stomach. In the stomach, peristalsis is a bit powerful because the wall of the stomach is thick and full of three layers of muscles, and therefore peristalsis there is called churning, and it has, as you know, a mixer-like effect to mix food with the digestive juices. In the blood vessel wall, it depends, the function of the smooth muscle, this is very important, will depend upon what kind of vessel you're talking about. If you're talking about the large arteries, then part of the function of the smooth muscles is to contract in order to propel the food forward. But if you, and therefore they are helping the pumping function of the heart. But if you're talking about the arterioles, and this is very important, the arterioles represent the last arteries before the capillary bed, because you have the large artery, you have the aorta, and then the large arteries, and then the medium-sized arteries, and then the small arteries, and then you have the arterioles. So the arterioles are the last arteries before the capillaries, and their main function is to regulate the blood flow to the capillaries and to the capillary bed, and therefore to regulate the blood flow to the tissues. Of course, regulating the blood flow to the tissues means regulating the oxygen that is delivered to the tissues, regulating the nutrients like glucose that are delivered to the tissues. But you have to describe this in detail. And you have to take care that you have to start by the function of the muscle, contracts or relaxes, and then how this is reflected on the diameter of the arteriole, and therefore on the flow. So you say that if these smooth muscles contract, then the arteriole constricts, then there is diminution of the blood flow to the tissues. We sometimes need to do this. And on the other hand, if the smooth muscles dilate, the smooth muscles relax, then the arterioles dilate, then the blood flow will increase to the tissues. So the arterioles represent what we call the peripheral resistance, the total peripheral resistance, because they can constrict and resist blood flow to the tissues or diminish blood flow to the tissues, or they can relax and allow blood flow to the tissues. But meanwhile, while controlling the blood flow, they also control the blood pressure, because whenever all the arterioles in the human body constrict, this meaning that the total peripheral resistance rises, this will elevate the blood pressure in people, of course, who are predisposed, like in cold weather or when the person is under severe stress or nervous. When the person is nervous or under severe tension. On the other hand, if all the arterioles dilate, meaning that the total resistance diminishes, then this will diminish the arterial blood pressure. Concerning the smooth muscles in the iris, they are very important. So you might be asked about the function of the smooth muscles in the iris, or you might be asked to describe how the smooth muscles in the iris regulate the light intensity that reaches the eye, or how they are adapted to carry out their functions. So as it is seen here, we have two types of muscles. We have the radial muscles and we have the concentric circular muscles. When the radial muscles contract, they cause dilatation of the pupil or increased size of the pupil, and this is what we call pupillodilatation. And this occurs, of course, when you are in dim light and you want to dilate your pupil to make up for the diminished intensity of light so that you can visualize the environment around you. But when you are in intense light, in great light intensity, then in order to protect your retina against this high intensity, the constrictor pupillae muscle or these circular concentric muscles will contract and diminish the size of the pupil and cause pupilloconstriction. So similarly to the arterioles, we use two verbs. We use contraction of the dilator muscle and pupillodilatation. Contraction of the constrictor muscle or the concentric muscle and pupilloconstrictor. But notice here that the difference between the iris and the arterioles is that in both cases of the iris, the muscles contract. When the dilator contracts, it dilates the pupil. When the circular contracts, it constricts the pupil. And this is how light intensity is regulated. I'll not ask you about the sympathetic and parasympathetic innervation to the iris. In the ureter, in the urinary system, of course, this is contraction of the smooth muscles is important to carry out peristalsis again and to wash the urine downward towards the urinary bladder. But we also have an important system, which is the respiratory system and the bronchial system, or the bronchi in that respiratory system that are lined with smooth muscles. And just like in the arteries, where the arterioles are the most abundantly supplied with smooth muscles, the bronchioles, which are the terminal smallest bronchi in the lungs just before the air sacs in the lungs, are the ones that are the mostly supplied or the most richly supplied with smooth muscles. When the smooth muscles in the bronchi, in the respiratory system contract, bronchoconstriction occurs, and there is diminution of the air that goes to the alveoli or the air sacs. On the other hand, if those bronchiolar muscles, which are smooth muscles, dilate, there will be bronchodilation and increased passage of air to the air sacs. This is a physiological process. This is a normal process to regulate the air that passes to the air sacs. But if bronchoconstriction occurs excessively, like in certain allergic conditions where there is an excessive, abnormally excessive reaction to certain antigens like dust, like perfumes, like pollen grains, and there is severe bronchoconstriction or actually bronchioloconstriction, then we can hear wheezes on the chest using the stethoscope, of course, when we auscultate the chest, and the patient would suffer from difficult breathing or dyspnea. Now, the characteristics of every type of muscle are very important. So the smooth muscular tissue consists of smooth muscle cells that are unstriated, that are tapered from both sides. That's why we call them, or we describe them as being spindle-shaped. They have, as every muscle cell has a single central nucleus. Of course, they have a cell membrane called sarcolemma. They have cytoplasm called sarcoplasm. This is if I ask you to describe the smooth muscular tissue under the light microscope. So if I ask about the structure, you wouldn't say involuntary. But if I ask you about the characteristics in general, then those characteristics might be structural characteristics like spindle-shaped, unstriated, and of course, unbranched and unconnected. But in addition to this, you have a functional property, which is that they are involuntary, that we cannot control their motion. When we say non-branched, non-connected, we are reminding ourselves that they are unlike cardiac muscles, which are branched and connected. So how many structural or histological characteristics do we have? Spindle shape, this is number one. Unstriated, number two. Unbranched, number three. Unconnected, of course, if they're not branched, they'll not be connected. And when we say non-connected, we're not talking about the connective tissue. We mean non-connected as muscle fibers through their sarcolemma. And the fifth histological characteristic is a single central nucleus, but the functional characteristic is that they are involuntary. Coming or moving on to the cardiac muscles, we also have functional characteristics, which are that they are involuntary, but we have structural characteristics. So they are banded or striated, they are branched, and they are the only type of muscles that are branched. And since they are branched, then they will be physically interconnected or structurally interconnected through, as you know, the intercalated discs, and every cardiac muscle fiber has a single central nucleus. But notice that the fact that they are interconnected will also result in a functional property, which is the fact that they can communicate together through the gap junctions in the intercalated discs. And the gap junctions consist of ion protein channels that allow the rapid movement of ions between the cardiac muscles, and therefore the rapid transfer of electric impulses over the whole myocardium that would act in such a case as a functional syncytium. A functional syncytium means that all the cardiac muscles contract almost simultaneously as if they were one muscle. But what does the word syncytium originally mean? The word syncytium originally means a big, large cell with multiple nuclei, which is not the case for cardiac muscles. The cardiac muscle structurally is not a syncytium, but functionally it is a syncytium because the presence of the gap junctions allows them to contract almost simultaneously because of the rapid transfer of ions. The skeletal muscles, functionally, they are voluntary, meaning that we can give them orders to contract on purpose. They are structurally similar to the cardiac muscles in many aspects because they are, or in just one aspect, actually, they are striated, but there is another aspect which is being cylindrical and elongated. That is not written here, but major differences between the skeletal muscles and the cardiac muscles is that the skeletal muscle is non-branched, and therefore the skeletal muscle is not connected, and there are multiple peripheral nuclei. And the presence, why, if you're ever asked to justify the presence of multiple peripheral nuclei, it's because the cardiac muscle, the skeletal muscle is a very active muscle, and the presence of multiple nuclei allow the metabolism of the muscle to be extremely rapid and powerful. Of course, the skeletal muscle fibers move body parts. As you can see, the biceps muscle, you can see here. Why do we call it a biceps muscle? Because it has two heads at the origin. The two heads are connected to this triangular bone that is called the scapula. It also has two small tendons at the insertion, one connected to the radius and one connected to the ulna. The biceps muscle is a skeletal muscle whose main function is to contract. But as we said before, organs carry out one or more interrelated functions. So what are the interrelated functions of the biceps muscle? The main function of the biceps muscle is to contract, but there is another important related function, which is flexion of the elbow joint. And flexion of the elbow joint, how is it interrelated with contraction? Because when the biceps muscle contracts, it will cause flexion or bending of the elbow joint. The biceps muscle, in order for it to cause flexion of the elbow joint, it has to stretch across the elbow joint. And this is one important condition for a muscle to move a joint. The important condition for a muscle to move a joint is to extend across that joint. Sometimes the muscle stretches across the joint, but in a position that does not allow it or that does not enable it to move the joint. So contraction of the biceps muscle, a strenuous contraction or severe contraction of the biceps muscle can lead to minor elevation of the shoulder joint because it stretches across the shoulder joint, but actually it cannot do this sensibly or in a significant manner unless it is held by other muscles around the shoulder, like the deltoid muscle, for example. In this diagram, we can also see that contraction of a muscle. I might ask you, justify contraction of the biceps muscle results in flexion of the elbow joint. This is because the biceps has an origin, which is the tendon that is connected to the stationary bone, which is here the scapula, and it has an insertion, which is the tendon that is connected, or the tendons that are connected to the movable bones. And when the biceps receives a motor impulse, it contracts, and when it contracts, actin slides over myosin, and there is shortening of the muscle. And when the muscle shortens, it is the insertion that will move towards the origin. And that's why it is the radius and ulna that are elevated to move towards the shoulder joint. So this type of question is actually a group of questions. Again, when I say why the biceps contraction results in flexion of the elbow joint, then you have to talk about sliding of actin over myosin. You have to talk about shortening of the biceps. You have to talk about the tendons that are at the insertion that move towards the origin. On the other hand, the triceps is the antagonist of the biceps. What are antagonistic muscles? Antagonistic muscles are muscles that do opposite functions, like the biceps and the triceps. For example, when the biceps contracts, it flexes the elbow joint. But when the triceps contracts, it extends the elbow joint. The biceps and triceps alternate, meaning that usually when the biceps contracts and flexes the elbow joint, the triceps has to relax in order to allow the biceps to contract. But when the triceps decides to contract or receives a motor order to contract and extend the elbow joint, the biceps has to relax. So the presence of antagonistic muscles around a joint allows the joint to do multiple functions, but at the same time, it should be taken into consideration that those antagonistic muscles do not hinder each other, but on the other hand, they coordinate together in order to allow smooth function. So here, this slide is talking about the origin and the insertion, and I've just mentioned the definition of each. And you have to be very careful because you always give distorted, or you usually give distorted answers. So the origin is the part of the muscle, or it is the tendon that is attached to the stationary bone, whereas the insertion is the part of the muscle or the tendon that is attached to the movable bone. So every time you have to start by it is the tendon or the part of the muscle that. The skeletal muscles that move the face are here just for your general knowledge to see how they are responsible for your facial expressions. And the only thing you have to know is that when you have paralysis of the facial nerve, which is the seventh cranial nerve, you will have this disease that is called facial palsy. Facial palsy is paralysis, lower motor neuron paralysis of one half of the muscle. And it is a lower motor neuron because it is an affection of the motor nerve itself, starting from the anterior horn cell in the brain, not in the spinal cord, because the facial nerve is a cranial nerve. It is the cranial nerve number 7, so it arises from the pons, a part in the brain stem, and therefore it is the nerve itself that is affected, maybe by a viral infection, maybe by whatever kind of inflammation that affects the nerve. You will have paralysis on the same side because it is a lower motor neuron disease. So what is facial palsy? It is a condition where the facial nerve, or the seventh cranial nerve, arising from the brain stem, is inflamed. It is the nerve itself that is inflamed and not the muscles. So it is a lower motor neuron disease, and it is a neuromuscular disease since the nerve is affected. Since it is a lower motor neuron disease, then you will have flaccid paralysis of half the face that is on the same side of the lesion, meaning that it will be ipsilateral paralysis. The patient will be unable to close the orbicularis oculi, meaning that even when the patient is sleeping on that side, the patient will be unable to, suppose that it is that side that is affected, he will be unable to close the eye, and you will need to put ointments and creams on the eye to prevent infection. When the patient smiles, This muscle is okay, for example, on the left side, where the facial nerve is intact, but that side is paralyzed, so the smile will be a bit weird because it will occur on one side only. And then we are going to talk now about the skeletal muscle fiber. As you can see, the muscle fiber or the myofiber is a muscle cell, and you can see that it is the skeletal muscle fiber is elongated and cylindrical, and it has multiple peripheral nuclei, and it is rich in mitochondria because the mitochondria are the sites of aerobic cellular respiration, and they are the site of producing energy. And it is the muscle fiber, or this is a muscle cell, so it should be surrounded by a cell membrane, which is called the sarcolemma. Of course, this picture is an electron microscopic picture because we cannot see the sarcolemma clearly under the light microscope. We cannot see the mitochondria under the light microscope, but we can also see them under the electron microscope. And embedded inside the sarcoplasm of the myofiber or the muscle cell, we can see smaller elongated cylindrical structures, spaghetti-like structures, which we call the myofibrils. And the myofibrils are the structures that are responsible for the striations of the skeletal muscle fiber. So if I ever ask you about the structure that is responsible for striations of the skeletal or cardiac muscle fibers, you would say the myofibril. Why? Because the myofibril is made up of myofilaments. So the smallest structure is the myofilament or the protein molecule. What are the myofilaments that make up the structure of the myofibril? They are actin and myosin, thin actin and thick myosin, and they alternate together to give the striated appearance. So the smallest striated structure in the muscle is the myofibril. And since all these myofibrils embedded in the sarcoplasm are striated, and since they are arranged in a parallel way, then they give the whole muscle this striated structure or banded structure. The diagram we have to the right here is a transverse section in a whole muscle, and that is the tendon of the muscle. And as you can see, the tendon of the muscle is the connective tissue cord that connects the muscle to a bone or to the corresponding bone where it is attached. But what I have to highlight here is that the epimysium, which is the connective tissue around the whole muscle, will taper to form the tendon. So the tendon is nothing but an extension of the epimysium. In the cross section of the muscle fiber, we can see multiple muscle bundles, and the connective tissue that surrounds every muscle bundle is called connective tissue perimysium. So the perimysium is what? It is the connective tissue that surrounds the muscle bundle, the whole muscle bundle. So every muscle bundle is covered by connective tissue perimysium, and the whole number of muscle bundles or the whole muscle is covered by epimysium. And every muscle bundle is nothing but a group of muscle fibers. Of course, all this is important in scientific terms. Again, here we have the myotendinous junction or the tendon, the connective tissue epimysium, the connective tissue perimysium. Of course, there is merging between here, for example, you can see merging between the perimysium that extends around the bundle and the epimysium that surrounds the muscle. So if I ever ask you to label this, you can say perimysium or epimysium, both will be correct. We haven't covered the satellite cell, so I'll not be asking about it, but here you can see that inside every muscle bundle, you have a motor nerve fiber and you have a blood vessel. And that proves that, as we said before, the muscle is an organ. Here are the characteristics of the skeletal muscles. And as we said, they are multinucleated. They have multiple peripheral nuclei, that every muscle consists of bundles and that every bundle consists of fibers. So there is an important question here, which is to arrange the structures descendingly or ascendingly. So if we start by a muscle, then we will have muscle bundles afterwards, and then myofibers or muscle cells, and then myofibrils, and then myofilaments. On the other hand, if I ask you to arrange them, this hierarchy ascendingly, you will start by the myofilaments, which are actin and myosin, and then the myofibrils, which is the striated structure, and then the muscle fiber, and then the muscle bundle, and then the whole muscle. And here we can see again the structure of a skeletal muscle fiber and why it is striated. So this diagram is an answer to the question that I've just mentioned a few minutes ago, which is, why is a skeletal muscle striated under whether the light microscope or the electron microscope? Because embedded in the sarcoplasm, we have the famous structures that are called the myofibrils. And the myofibrils, as you can see, consist of sarcomeres, and the sarcomere is the structural and functional unit of the myofibril and of the whole muscle. And inside every sarcomere, or on both sides, we have Z lines. So you have a Z line here and a Z line there. So the sarcomere is the functional unit of the muscle, and the sarcomere is bounded on both sides by two Z lines, to which are connected the actin filaments, and alternating with them, we have the thick myosin filaments. The areas of the sarcomere or of the myofibril, as you wish, that contain both myosin and actin are the A bands or the dark bands, whereas those areas that extend over two sarcomeres that contain only the actin filament, like this part, for example, are considered the light bands.